Hello, my name is Danielle Flores Lopez. My pronouns are she, her. I serve as the interim assistant dean for diversity, equity, inclusion, and the director of academic advising and student success initiatives in the College of Natural Science. We're going to start with the land acknowledgement. We collectively acknowledge that Michigan State University occupies the ancestral, traditional, and contemporary lands of the Anishinaabe, Three Fires Confederacy of Ojibwa, Ottawa, and Potawatomi peoples. In particular, the university resides on land ceded in the 1819 Treaty of Saginaw. We recognize, support, and advocate for the sovereignty of Michigan's 12 federally recognized Indian nations, for historic Indigenous communities in Michigan, for Indigenous individuals and communities who live here now, and for those who were forcibly removed from their homelands. By offering this land acknowledgement, we affirm Indigenous sovereignty and will work to hold Michigan State University more accountable to the needs of American Indian and Indigenous peoples.
Good evening. My name is Tasha Warfield. I'm the academic advisor over in Lyman-Briggs College. I use the pronouns she, her. On behalf of the MSU Amplified STEM Planning Team, which consists of the College of Agricultural and Natural Resources, Lyman-Briggs College, the College of Natural Science, and the College of Veterinary Medicine, we welcome you. As we know, STEM is a term used to group together 4 major areas and dozens of academic disciplines. Although each discipline stands alone, there is value in them being recognized together. As we, a collective of science-focused colleges, come together to amplify diverse voices in STEM, there's power in us working together. And as we seek to uplift diverse scientists and amplify their work, we know that diversity does not simply encompass various ethnic and racial backgrounds. This LGBT History Month, we celebrate the STEM contributions of people of all genders and sexual identities. We also recognize that many voices of LGBT folks in STEM have gone unheard. To amplify diverse voices and contributions, the planning team continues to work diligently to bring you events. We hope that each program is a learning opportunity to become more knowledgeable so that you can help us all amplify STEM contributors.
Good evening. My name is Diana Bello de Ocampo. I'm a professor with the Integrated Biology Department with the College of Natural Science. My pronouns are she, her, hers. It is my pleasure to give a brief history of LGBT Month. Lesbian, Gay, Bisexual, Transgender History Month was created in 1994 by Rodney Wilson, a high school history teacher in St. Louis, in collaboration with educators across the United States. They selected the month of October to correspond with the National Coming Out Day on October 11th, and to commemorate the anniversary of the first March on Washington for LGBT rights on October 14th, 1979. LGBT History Month was endorsed by national organizations including Gay and Lesbian Alliance Against Defamation, GLAAD, the Human Rights Campaign, the National Gay and Lesbian Task Force, and the National Education Association. In 2006, the nonprofit Equality Forum created LGBTHistoryMonth.com, a website that highlights 31 diverse icons annually to create a national conversation on LGBT history. History Month is dedicated to recognizing and celebrating the history and achievements of LGBTQIA2S+ community members and their fight for freedom and acceptance.
We're so pleased to have Daniel Pfau joining us tonight for a research presentation entitled Moving Beyond the Biomedical Binary, Mouse Models of Gender-Affirming Hormone Therapies. My name is Kendra Pyle. My pronouns are she, her. I am the Diversity, Equity, and Inclusion Coordinator here in the College of Natural Science and also a graduate student in the School of Social Work.
I'm very pleased to introduce Daniel. Daniel Pfau, PhD, they, them, their, is a queer, intersex, and genderqueer neuroscientist specializing in endocrinology. They earned their PhD in neuroscience here at Michigan State University while receiving support from an NSF Graduate Research Fellowship and an MSU University Enrichment Fellowship. Currently, they are a postdoctoral researcher at the University of Michigan studying mouse models of gender-affirming hormone therapy with Drs. Moravec, Shiganov, and Padmanabhan. They have spent 15 years increasing diversity, equity, and inclusion in STEM and academia through advocacy and education. Outside campus, they have held positions on a community pride boards and co-founded Queering Medicine, a capital area grassroots organization seeking to address the healthcare needs of LGBTQIA+ residents. They have been featured on Ologies with Ali Ward, Pride podcast with Levi Chambers, and write a blog for Psychology Today. Dr. Pfau is also the parent to 3 amazing children, a partner, an avid reader, and a gardener. Welcome, Dr. Pfau. I'm going to turn it over to you for your presentation.
Thank you so much for everyone providing those wonderful introductions and information. I am going to get my presentation shared. And it should be up. Okay. So thank you everyone so much for coming today, and thank you so much for the organizers for inviting me. My talk today is entitled Moving Beyond the Biomedical Binary, Mouse Models of Gender-Affirming Hormone Therapy. And just to give you a little of an outline as I go through of what I'm going through today, first I'm going to give some background both on gender-affirming hormone therapy and also what I mean when I say beyond the biomedical binary and mouse models of gender-affirming hormone therapy. Then I'll be going over some of the current projects that I'm working on, including one study looking at estradiol therapy and another looking at testosterone. And then I'll offer some conclusions across these studies. So first of all, the gender-affirming hormone therapies, or GAHT, are therapies that the transgender and gender diverse community may utilize. And it helps individuals to achieve embodiment goals. And we know that it improves mental health, and it is a life-saving intervention for some individuals. We also know that it's safe and effective, meaning that these individuals are going to get the outcomes they're expecting without having it impact their daily lives, aside from the mental health improvements that they see. Still, we know that the GAHT, I'm going to be calling it from now on, has widespread effects. And one of its main effects is on the natal hypothalamic-Pituitary-Gonadal axis, or HPG, and it really impacts its function. So gender-affirming hormone therapies alter the circulating steroid hormones. Typically, we have steroid hormones being produced by the testes and ovaries that go to the brain, and they provide a signal here that tells the HPG how it should react to different, or it tells the HPG to turn off and on. And so the hypothalamus communicates with a specialized population of neurons that released gonadotropin releasing hormone. And the gonadotropin releasing hormone travels through the blood into the pituitary. And the pituitary is where luteinizing hormone and follicle stimulating hormone, the 2 gonadotropins, are released. Those travel to the ovaries and testes, where those steroid hormones are produced, and the feedback system continues. As I said, GAHT, or GAHT, is going to be the introduction of an external steroid hormone source. And that introduction leads to a constant negative system, or negative signal on this system. So GAHT suppresses the gonadotropin production that's a result of GnRH from the hypothalamic region. And then because those gonadotropins are lost, it prevents the steroidogenesis, so the production of the steroid hormones within the gonads. We really don't know exactly how this happens or whether or not this has long-term effects on overall health. And I think one of the groups that is truly driving the need to look into this is the transgender, gender diverse, and non-binary community itself. So this is a quote from a study that's been going on where these researchers go and ask community members to sit on panels and provide sort of feedback on how they feel or what they feel are research priorities. And so this is just part of the quote. We need research on the drugs we take for years and years and years. And just to highlight some of the issues these individuals are bringing up are the importance of long-term research on GAHT, the lack of clear answers that they're receiving, how many unknowns there are, and that there really is just no research. And so GAHT mouse models offer an effective alternative to the fact that the controlled research that we really want to be able to perform to understand GAHT cannot be performed in a population where that intervention is medically necessary. And so, as I said, mice offer an effective alternative, and they can do this and they allow us to study a number of different topics that have been identified by the trans and gender diverse community. For example, its impact on reproduction, the long-term health effects, and possibly looking to improvements for the regimes. So I keep mentioning these GAHT models, but really what are they? And to discuss that, I'm gonna sort of bring us to the idea of this biomedical binary. In classic endocrinology, where we find people studying in the field of sex differences, we have these classical comparisons. So one of them obviously being at the top here, the gonads of a male and female. What we're basically saying is that oftentimes to study sex differences, we'll compare animals based on what gonad they have. Another thing that sex difference researchers do is gonadectomies, where they remove the organs producing the steroid hormones, so that would be the gonads, but then they replace those steroid hormones, either estradiol and progesterone or a testosterone supplement. And so we know we can make these manipulations, and it's easy to sort of see and take these and do the same things, but to reproduce what we do when providing testosterone GAHT or estradiol GAHT. And so as you can see here, rather than just comparing between mouse who has ovaries and testes, we can include another group where we have a mouse who has ovaries but is being treated with testosterone. Conversely, we can do the same looking over here at pubertal GAHT, where we have intact animals that have, for example, ovaries, but others where we provide a GnRH agonist to them. And so each of these comparisons can provide valuable information. And I'm going to be focusing today, though, on a TGAT model and an EGAT model. So let's stop for a second. Did I just say there's something wrong about studying sex differences? And no, that's not what I believe. In fact, I believe that sex difference research is essential to equitable healthcare by providing information on a diversity of bodies, and a diversity of bodies with multiple different kinds of hormonal milieus is also important. And I believe this is made difficult because we need more inclusive language. And so one of the things we have to recognize about sex difference research, and I'm going to be sort of taking apart this diagram on the right now. I pulled it up off of Wikipedia, but I think it really explains and shows some of the assumptions that are made. And so the first one is we use unnecessarily gendering language. So I just removed some boxes that were covering up the words male and female above testes and ovaries. And that's superfluous. We don't call things male brain because it's in a male, we call it a brain, and we analyze different brains versus males and females. The testes themselves can exist outside of the male body, and so can ovaries. And being able to have that language to be able to, just say testes allows us to discuss a diversity of individuals who may have testes. It's also context dependent. So as you can see, I got rid of the steroid hormones, and this diagram originally said testosterone, estradiol, and progesterone, separated by where they're coming from. However, we do know, despite the fact that testosterone is higher in the male individuals we might want to be testing, there are impacts of estradiol and progesterone that we might want to research. So whether or not testosterone is the main impact within a certain body doesn't preclude the fact that we need to look at other aspects of what are considered sex differences. And then finally, it's multidimensional. So we have several levels here looking at, for example, gonad presence versus presence of different hormonal milieux, all the way up to hypothalamic differences based on the developmental exposure to hormones that an animal might receive. And so all of these things, when we are intentional about them, they allow us to not only be more accurate in the way that we describe sex difference research, but also more affirming as we can describe more bodies and more diversity within sex and gender. So I'm going to go now on to how I have sort of done this by trying to look at two different GAHT mouse models and sort of using the knowledge that I've gained through my education in sex difference to sort of shape our language and shape the way that we look at how transgender and gender diverse communities benefit from science. So the first one I'm looking at that we'll be talking about today is the reproductive and hormonal impact of estradiol gender-affirming hormone therapy. So for this one, I have 4 different treatment groups, and it just so happens that no one has actually looked at a mouse model for reproduction, meaning no one has taken a mouse that's intact, still has its testes, and treated it with estradiol to see how the reproduction of that animal will be impacted by the long-term effects of GAHT treatment. And so here I decided to include 3 different dosages because we were not sure which one would lead to the effects that we wanted, and also a control group that was basically implanted with a capsule that was left blank. And so just the sort of diagram here explains how we did the study. Before the animals were given the treatment, we collected some blood and took images of their genital regions. After we gave the implants, we allowed the treatment to go on for 6 weeks, during which we collected blood and took 1 more image of their genitals before taking terminal images of the genitals, collecting terminal blood, and then we took their tissues, their gonad tissues, and did hematoxin and eosin staining to analyze histology. And so for the results from this study, we found that GAHT does alter the genital anatomy of mice. And so we were able to see that the external presence, or the visible presence of an epididymis within the scrotum was lost across the study. So animals given GAHT by the end of the study could no longer you could no longer discern the sort of pocket that the external or the internal epididymis structure would be forced into within the scrotum. And I will have images of the internal structures later on too. And so the yellow arrows here indicate where, for example, in the blank, we can see that there's these 2 protrusions. And then also in this 1.25 milligram animal, we can see that only one side of the, or one testy has an epididymis that is protruding while the other one seems to have receded. In addition to these changes, we found that the preputial sheath actually changed elasticity. So in blank animals, when you pull the preputial sheath back, and I was doing this to image the penis, the preputial sheath would automatically recover itself and the penis would retract. In the animals that were treated with estradiol, by the 3rd week, we found that the retraction of the sheath required then us to use external pressure to return the penis. And so in this diagram, you can see that these look typical to the blanks, but it is only when you push it back and release it that you can see that the sheath won't recover itself. So in addition to those external effects, we examined the hormones across the study. And so before we implanted these animals, the testosterone levels were equal. But by the time that we were about a week out from the implants, our group given the 5 milligram of GAHT had almost a trend towards lower testosterone levels. By 2 weeks after, all of our groups had significantly lower levels of testosterone, and that continued to the end of the experiment. Looking at terminal levels, the steroid hormone levels were following the profiles we expected, testosterone being decreased in our GAHT animals, and estradiol being increased in our GAHT animals. We also found that we're able to suppress the HPG's gonadotropin production Both the follicle stimulating hormone and luteinizing hormone levels were decreased, although the luteinizing hormone was only significantly decreased in our highest EGAT dose, the 5 milligrams. And this is interesting because we do see in clinical populations, oftentimes individuals who have an HPG that is resistant to suppression by estradiol alone. And as I indicated in the diagram above, sometimes progesterone and other treatments called antiandrogens may be included to address this. Looking at the androgen-sensitive tissues internally, the testicles and epididymis showed some striking changes. Right here at the bottom of the testes, in each of these images, you can see the epididymal structure that is located at the portion of the scrotum where you could see sort of that protrusion. As you can see, we do see a decrease in the amount of tissue there. And then also looking at weights, we do see within the 5 milligram a significant decrease. Looking at the testicle weights themselves, we see a dose-dependent effect. But in general, we see that the testicles are lower in our EGAT animals. Looking at another tissue that is highly hormone sensitive, the seminal vesicles, we do see that EGAT treatment severely decrease the weight of these organs, which we would expect. An interesting finding, however, was that looking at bladders, we found an increase in the weight of those from our EGAT treated animals, and it was once again dose dependent. There is anecdotal evidence within EGAT patients of issues related to bladder, but those have not been studied in depth, and this provides some evidence that maybe there is something to be looked into there. Then looking at the histology of the testes, we found that EGAT does disrupt spermatogenesis. And so looking at images in the top panel here, we can see that within the blink animals, the spermatogenic cells within the seminiferous tubules, those circles there, extend all the way into the lumen, whereas in each of our EGAT treated animals, we can see varying degrees of what's termed vacuolytic tubules. So each of the arrowheads points to a seminiferous tubule where some of the germ cells have been sloughed off and it's left this sort of vacuolytic morphology. And then the bottom panel, the first two images are showing have arrowheads showing that the Leydig cells are still present within our EGAT treated animals. And then those final two panels on the bottom are showing some of those germ cells that would be on the edge of the seminiferous tubules being sloughed off and creating these vacuolytic morphologies. And we did find that the number of vacuolytic tubules differed by dosage. And so those with 5 milligram treatments had the highest number of these vacuolytic tubules. When we looked at the actual sperm within the epididymis itself, though, we found that there was mature morphology as well as immature morphology in all of our groups. Yet in our EGAT treated animals, the locomotion of those sperm was disrupted and they only showed a shaking phenotype rather than showing the capability of swimming. Very interestingly, we took a look at the relationship between this, our marker of spermatogenesis, which was the vacuolytic tubule number, and we decided to see if we can see any relationships with the terminal hormone levels we collected. And while the gonadotropin follicle stimulating hormone is typically thought of as being involved with spermatogenesis, we found that it was luteinizing hormone levels that were negatively associated with the vacuolytic tubule numbers. Looking at our steroid hormones, however, there was no impact. Now moving on to the epididymis histology itself, we see within the caput epididymis that there isn't many changes to the lumen of these tubules, the epididymis tubules, but the tube area itself, so the area taken up by the tube itself here, as is measured on the cross-sectional area, was decreased within our EGAT treated animals. We also took a look at the cauda epididymis, and as you can see here, one of our findings was an increase in this interstitium medium that you can see within each of our EGAT treated groups, and it definitely increases in our highest dosage. But we also find the opposite relationship within the tubes here. So the cauda lumen is what is decreasing in size within our EGAT treated animals, while the tube area itself remained the same. So just to go over the actual comparisons to the clinical outcomes, I'll go over a few here and highlight some that are really important. So first of all, obviously with the testes, we saw the lower weights. And then interestingly, within our study, we found that there were still Leydig cells present, although in some of the clinical data, these show absence in some individuals while are completely present in others. And a few of these other measures, including spermatocyte presence, as well as LH hormone levels, can show high variability between patients. And so the reason why LH might be important is because, for example, an EGAT patient might find that the estradiol they're taking is not sufficient to suppress their hypothalamic-Pituitary-Gonadal excess. And being able to use a mouse model where we have different dosages inducing different suppressions of the HPG, we can add anti-androgens in and see how their effects might differ. In addition to sort of this interesting aspect in relation to the clinical data, we also were able to show that the high variability within spermatogenesis in the clinical populations might be related to the levels of LH. And this is not only supported by our work, but other work showing that in trans women, the marker MAGE-A4 is positively associated with LH, and that's a marker of spermatogenesis. And then, like our study, another found that testicular atrophy is negatively associated with LH. So being able to look at that relationship is a really interesting aspect of our outcome of our study. So now, before I go into some overall conclusions on sort of these GAP models, I'm going to talk a little bit about another study that I've engaged in, which is the impact of testosterone on clinically relevant brain nuclei in mice. And so this model is actually following a lot of research that's happened in the lab already. So TGAT mice, the publication that shows sort of these outcomes in the TGAT mice has already gone out, and several other studies have gone on in the gonads. And so just to give you an overview of the findings from those, first of all, in female mice and in TGAT mice, we're able to track their vaginal cytology. And that basically is collecting cells from the vagina and taking a look at them under a microscope. And as you can see in the left panel under controls, there's estrus, perestrus, diestrus, metaestrus, each of these showing different cellular components. When we look at a T-treated animal, after we begin the T treatment, all these animals show a persistent diestrus, indicating acyclicity and disruption of the HPG. So these graphs over here are just a representation of that same data, showing that after the T treatment began, we see this line that just goes on diestrus. And when we look at the testosterone, when they've looked at the testosterone levels, they do see the increase that they're expecting. And they also see a decrease in LH that's evident in the clinical population. The external anatomy shows similarities to clinical populations as well, in that TGAT mice have clitoromegaly, an increase in the size of the clitoris. And their ovarian phenotype is similar to polycystic ovarian syndrome, which is what we also find in TGAT patient populations. So for example, in these left two images, you can see that the control animals have corpus luteum, indicating that they are going through the estrous cycle, while both T-treated animals do not have these corpus luteum. So using the TGAT model already developed in the lab, I decided to look beyond the gonads and to the brain. And so for this study, I had two different groups. One based off the previous research, we used a 10 milligram testosterone capsule, and then a blank untreated control group. And what we were hoping to take a look at was the chronic effects of gender-affirming hormone therapy on some regions in the brain that are related to the HPG and clinical evidence. So to do this, we started by looking at the daily vaginal cytology, as well as collecting blood from the animals to ensure that we had the similar physiology at the beginning. Then after the implants were introduced, we continued vaginal cytology until we saw that persistent diestrus typical of this model. We collected blood from the tail in order to ensure that we were seeing the the increase in testosterone levels brought on by the treatment. At the end of the experiment, the animals were perfused with paraformaldehyde and the brains were post-fixed. They were sectioned, and one of the three series that were taken from them was stained with Nissl stain, and I'll go over that in a little bit, which was then analyzed with ImageJ. So our Nissl analysis was done in 3 different brain regions, and these were chosen based on the clinical data available and their association with the HPG. So first, the bed nucleus of the stria terminalis is analogous to the region of the same name in humans. And interestingly, we do have clinical data, although the N for each of the studies, the BNST and the ARC, is an N of 1. So a transgender man has a larger regional BNST volume and more somatostatin neurons in his BNST when compared to the cisgender women in the study. And then in another study, it was found that the infundibular nucleus, which is in mice, the arcuate nucleus, had fewer kisspeptin neurons in a transgender man compared to the cisgender women in that study. We also included the anteroventricular periventricular hypothalamus, which is a region that's involved with the HPG, very similar to the arcuate nucleus. And in humans, it's the RP3V that's analogous to this, though it has not been examined within clinical populations of GAHT patients. So here you can see an image of the Nissl stain. Each of these blue circles is a cell and their specific morphology that's associated with neurons. And so I took these images wherever I could find that they aligned with my region of interest and didn't overlap with another image taken, and they also didn't extend into the other nuclei surrounding that area. And then I took each of these images and chose from them randomly 5. And all of the neurons in each of those images were counted to estimate density, and then the soma of 25 neurons within each were measured. So looking at our data collected across the experiment, we did find that our TGAT treatment led to the persistent diestrus we're expecting. So these are just that graphical interpretation of that data again, where we can see the TGAT animals persistent in their diestrus after treatment. We also found that it increased testosterone as we were expecting, and also decreased the LH levels as evidenced by the terminal LH we gathered. So then when we looked at the brain, we did find some significant impacts of the TGAT treatment. So looking first at the bed nucleus of the stria terminalis, we found that the number of neurons we counted in the frames was higher in the animals that were receiving the TGAT treatment. However, the soma area within this region was not altered by TGAT. Looking at the ARC, we found that the measures we were looking for were actually independent of GAHT treatment. So both the soma area and the number of neurons counted were similar between our treatment groups. Then looking at the anteroventricular periventricular nucleus, like the BNST, we saw a significant impact. However, this time it was on both the neuron soma size and neuron density. For soma size, we found that control animals had smaller soma than our TGAT treated groups, while neuron number was increased in the animals who were given the blank as opposed to those given TGAT. When you look at the sex difference research, interesting because when you look at male and female mice, this relationship has shown that it's been swapped. So in male mice, it's typical for them to have a higher number of, a lower number of AVPV neurons, while females are supposed to have a higher number. So what this shows us is that TGAT, like it alters the gonads, alters the brain nuclei that are clinically relevant. And just to go over those again and sort of compare the data clinically to that which we've collected. First, we have the BNST, which is larger in trans men and has more of the somatostatin neurons. And we had no effect on the size of neurons, but we did see that there were more neurons in the BNST of TGAT treated mice. Unlike the infundibular nucleus, we saw no impact of TGAT on the ARC. And then finally, looking at the AVPV, we did see something within our mice, the larger soma and fewer neurons. However, this has not been examined, as I mentioned earlier. And I do want to mention that it's very important. In order to make direct comparisons, we must include cell type specific analysis. So looking in the BNST, I'm hoping to examine the somatostatin number, and then obviously in the ARC, the kisspeptin neurons. And so just to get bring all this together and sort of come up with some conclusions. What I've sort of discussed today is that there are multiple GAHT mouse models and that these replicate the patient outcomes that we see across the HPG from the hypothalamus to the gonads. And the findings that we are getting from these mice are quickly increasing the number of questions that we might be able to ask to address the needs of individual patients. So for example, in regard to family planning, do the LH levels that we're seeing influencing spermatogenesis and EGAT patients explain the variability within clinical populations? For long-term health needs, do the structural changes that we see within the bed nucleus of terminalis, which is highly involved in metabolism, alter that? Dosage considerations. Why is e-treatment alone insufficient to suppress HPG in some patients, but capable of doing so in others? And understanding HPG suppression itself, how that process occurs. So, for example, are the anatomical changes to the AVPV within the TGAT mice, are those required for GAHT-dependent suppression of the HPG? And then I just want to, in addition to sort of talking about how this model can benefit the researchers that want to use it, but also the community, I think it's very important that researchers who engage in this type of research and individuals who want to learn about this type of research are also aware of the transgender and gender diverse communities' lived experiences. And so I included this final diagram, and this is from a recent publication by me in addition to several other transgender and gender diverse authors. And I'll include their names at the end, but each of these are just advice on sort of what you can do to educate yourself and improve the inclusivity of this research. So increasing the number of trans individuals engaging, ensuring that trans individuals know of opportunities to be hired into these kinds of experiments, ensuring that you cite transgender researchers who are working on this, and bringing those individuals like the amazing folks here have done to discuss these types of topics. And I think that sort of being very intentional about these aspects is important, given the fact that the transgender community has experienced prior harm due to interactions with the biomedical community. And so these key considerations can really ensure that the products that are made by scientists are well received, not only by the scientists around us, but by the transgender and gender diverse community itself. And so with that, I have nothing else for you. I just want to say a big thank you to so many people. So first, my 3 PIs, Molly Moravec, Ariela Shikhanov, and Basanta Padmanabhan, as well as the other folks working on our project, Cynthia De La Cruz, Amanda Schwartz, Hadrian Kinnear, who's now left our lab, but still continues in spirit, and as well as Jameson Clark, who's my undergraduate assistant. Many of the Shikhanov lab members, as well as I mentioned, the members of, or the co-authors on a paper I wrote recently, which included those suggestions that I had at the end. So Krisha Agi, Simone Sun, Teddy Goetz, Troy Ropke, and Eartha Makeup. And I'd also like to thank Kendra Pyle, as well as the NatSci DEI Associate Dean's Office, as I know they are the folks who certainly pushed for me to be coming back, and I am so very thankful for them to always be thinking of me. And as well, thank you to the Amplify STEM sponsors, all of the different colleges, natural science, I can't read it because this agriculture and natural resources, Lyman Briggs College, the College of Natural Science, and College of Veterinary Medicine. So thank you. And I also am very happy to note that in a little bit, you will be hearing a call to be supporting some local efforts to improve, the daily lived experiences of your local LGBTQ community. And so just harking back to, what I was talking in my last few slides, I'm just so glad that that is something that the organizers recognize is important. And with that, I guess we'll go to questions.
All right. Thank you so much for that, Daniel. So now this is an opportunity for those of you in the audience to ask some questions. And so if you want, you can post questions in the Q&A box if it's available for you. I also see that there's a couple of folks who have raised their hands. And so I'm going to allow them to speak. So just give me just 1 second here. If I can find, I think they lowered a hand. All right. And I just lost the window. Hang on. All right, and so I have one question here. Let me allow, I think it's Samin.
Hi.
Hello, how are you?
Thank you so much. Good, how are you?
Good.
Thank you for such an informative talk. I think it was too much to digest for me, but I have many questions, but I want to start with, how do you, Daniel, define sex through the lens of neuroscience?
Yeah, thank you for the question. That is a rather complex question. And I did bring up a rather briefly in one slide, I had mentioned a study that's out in science, or not a study, a letter that's out in Science, and how it describes sex in multiple ways. And as I had put it, it's a sex-dependent measure. It's context-dependent, it's context-dependent, as well as sort of the language within neuroscience itself has sort of limited our ability to discuss that. And so I do feel like while we have definitions for sex and use them within like our research papers, oftentimes, for example, we'll say something like a male body, blah, 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 blah. But when we are saying male body, that in one study could mean an animal who has no gonads and high testosterone, while in another study, an animal that still has its gonads, but we don't really actually know their levels of testosterone. And so when I say that, sex and defining it is a bit complex, I think it's just something that when sex difference researchers are discussing these things, they just have to be intentional and allow the complexity that is present to be part of the language, rather than just trying to simplify things to just a binary in order to be able to simplify it. And so while I don't necessarily feel that like I can define sex in so many and very simple words, I do feel like it's a conversation that should be happening in neuroscience. And, as you said, it's sort of a lot to wrap our heads around. And even for me, as I enter into like reading the literature at times, I'm like, okay, well, how could this be better phrased so that it, is more accurate and more affirming? And so as this field grows, I do believe that more neuroscientists will come into this conversation and sort of expand their idea of what can and what is required to define sex.
Thank you so much.
Sorry, that was a rather long answer.
No, thank you. I was just a little bit confused. Do you also see that as a spectrum? or is there any limit on like moving towards the ends of the spectrum, or it's just, as you said, depends on having some signs of being male or female, or like ovaries or testes?
Yeah, and I think because we have such an elongated development, there's so many opportunities for, minute variables to occur that might have effects farther down. And so we know during development, for example, there are periods when steroid hormones are high and periods when they're low. And that is one aspect that we can talk about, the fact that, the levels are there, but we can also talk about the fact that like when those levels appear. So if they're shifted in a certain direction, that might impact someone differently. And so when we have individuals who, for example, have most of their sexual development occurring, but for example, intersex individuals may not have complete sexual development. And sometimes that might be related to the fact that rather than not having or having too much of a hormone, they simply had it at a different time point. And so when we talk about, what sex is, if sex can vary based on when testosterone is high, as just as much as whether testosterone is high, then we have to accept that there's going to be more than just one outcome. And I think one of the important things to remember is within the field of neuroscience, a lot of research is coming out showing how hormonal history of a cell, so the history or the hormones that a cell has been experiencing throughout its history in life can truly impact its future function. And so variability within an individual or between individuals in hormone exposure, is going to lead to vast differences in neuron function based on the fact that these neurons are, receiving a whole life of information rather than, just having it during development. So I think sort of expanding past development into adulthood, seeing how hormone levels themselves are impacting things versus the time the hormone levels are there. I just feel that there is a lot that can be looked into and a lot that makes it not just, I think to me, it just makes it more interesting and, it's much more fascinating than simply these two outcomes.
Thank you so much. I do have more questions, but if anyone else wants to go ahead and ask question, I can ask later if you have time.
Awesome. No, yeah, we do have 2 more questions on the floor. So thank you. So what I can do is I'll take those, and then if we have a little bit more time, I can bring you back. Does that sound good?
Yeah, sounds awesome.
All right, awesome. Okay. Okay, so we have one question here in the, looks like 2 questions. All right, so the first one is, great talk. Do you see any changes to other hypothalamic nuclei involved in sexually dimorphic responses, such as the VMH, e.g. changes in lordosis, stress response, feeding behavior, etc.?
Okay, I'm just making sure I'm not muted. Yes. So I have not taken a look at those brain regions, but the behavioral responses associated with those regions are altered. So for example, the sexually dimorphic responses related to the hypothalamus, when you treat a female mouse with testosterone, we know that they will express same-sex mounting behaviors. And so when we see those in our TGAT mice, that's an indication that it's having an effect on that brain region, but we do not really know that whether or not that effect is structural, functional, or anything else related to, or what those specific changes are, but they do lead to a difference.
All right, and so thank you so much for that. And then we have another question here. do you have any data on how mice treated with GAHT respond to any other medication as compared to control? Basically, do medications work the same way as expected on GAHT-treated animals as compared to the control?
That's a great question, and that is, in addition to sort of examining GAHT's impact on the HPG, a topic that many trans folk want to hear about. And so, for example, we know within sex difference research that a lot of aspects of mental health are influenced by hormones, but not only the aspects of the mental health itself, but the treatment options for individuals. So I had talked before about, there's been a lot of research out on postmenopausal women and how steroid hormones might impact depressive symptoms then. And so, for example, in postmenopausal women, some of them respond to estradiol, or their depression responds to estradiol, while others don't. And so even within cisgender populations, we know there's variability based on hormone levels. And so it's definitely a possibility that when individuals are on gender-affirming hormone therapies, that it might first alter the way drugs are interacting with the system, or alter the systems themselves so that drugs interact with them differently. And so that's a great question, and I definitely believe that there are impacts, and I would love to start that kind of research.
Awesome, awesome. No, thank you so much for that, Daniel. And I do have another question here. So obviously, I mean, I'm sure that you've probably encountered many challenges, you know, as you've gone through this research, you know, probably a lot of successes as well, based on everything that you've shared with us. But, you know, I guess if you had to name one thing, what would be something like the most prominent thing that surprised you in all of this as you kind of ventured, as you started this journey in your research?
Yeah, I think one of the things that surprised me was how much we sort of hold on to the idea that the binary is important in some way, that, you know, that keeping the idea that we just need to look at two conditions is in some way gonna, you know, protect us from some sort of damage or harm, when in fact, it doesn't preclude the idea of analyzing these two different conditions. It simply expands this field into other arenas. And so oftentimes I feel as though there's sort of this pushback based on a need either to see expanding this as harmful to the research that's currently going on. So for example, sex difference research saying that, you know, it's going to take away from, you know, the people who would want to be doing that. But I mean, this is what I want to do, so I'm doing what I want to do. But then also I feel as though it sort of happens in the opposite way, where we have individuals who feel as though this binary informs science and really should be guiding our research. And it just seems so strange to me, because that's really saying like, oh no, we did understand everything, and so we don't really need to change our opinions. But the more and more we know, the more we understand that this variation is important, and whether or not, you know, we can fit it into this perfect binary of gonad versus gonad, gamete versus gamete, doesn't really matter when it's important to our health. And so I think that just sort of being able to move beyond that discussion and acknowledge, as I said before, that, you know, using more accurate language is also using more affirming language. And it's hard for some people, because I feel as though they want to, you know, be accurate, but they also don't want to be affirming, which is unfortunate. And so, yeah, I feel like there's a lot of variability within that sort of arena itself. And definitely I feel as though that has been one of the more difficult aspects.
So thank you so much. And it looks like we're kind of running near the end here. So, so with that, I want to kind of start bringing us to a close so that we can get everyone out here on time. And first and foremost, we would like to thank everyone for attending the Amplify STEM event tonight. Obviously, a big thank you to you, Daniel, for sharing this informative talk on how mouse models inform medical decisions related to gender-affirming hormone therapy. So we really appreciate that. And then also, I just want to say thank you to the committee members from all of the colleges who have been a part of this process and making sure that we put this together. And then lastly, I just wanted to drop a few resources in the chat as a call to action for local LGBTQIA2S+ organizations that you could support. And so I'm going to go ahead and drop those in the chat. And then with that, we're going to go ahead and bring things to a close. And thank you, everyone. And thank you, Daniel, again. And we wish everyone a good night. So thank you.